Assalamualaikum warahmatullahi wabarakatuh. My name is Ahmad Azam bin Abdul Jamil. I'm the project coordinator of our project. Our project title is Machine Learning Application Modern Approach to Evaluate Watermelon Quality. So, we want to assess watermelon quality. The problem beforehand is we use a traditional method such as visual inspection, tapping watermelon, or checking for spots on the fruit using our eyesight. But this approach has much noise and unreliable because they are prone to human error, and if we want to perform it on a larger scale we need an extensive labor and such on. So there also like problem with the traditional image recognition. According to Yuan, he says that the traditional image recognition require a very tedious step and issue which require the researcher to further their study, and this will result in require much longer time to obtain a robust accuracy. So we need a consistent and effective with a scalable solution that is able to identify watermelon quality based only on the inspection of the watermelon surface and leaf to classify the watermelon into such qualities such as color, spot, size, and shape. So the data set that we obtain are mainly coming from RoboFlow, and then we move to our objective. Is the first one is to assess watermelon quality using multiple techniques from machine learning that are suitable for our chosen dataset. And then number two is to implement machine learning into a real world dataset and evaluate it using standardized metrics. And the last one is to get a valuable insights of machine learning capability of solving real world problems.
We move to our first methodology, which is image grading classification. We have two methods, which is image processing and also random forest method. For the image processing, we use RGB value to extract the dominant color of our dataset. We will upload our dataset into a Python script using and we use and also use OpenCV library and K-mean from Scikit library to extract the dominant color and then store it in a CSV file. So as you can see here, this is the CSV file and the dominant color, as you can see here, the green is much more dominant than red and blue. So I store all of the data in this RGB value. Then we also have random forest method applied to our dataset because our dataset is small to middle size, and also that random forest has a huge resistance to overfitting issue other than other method like decision tree and so on. Furthermore, we use random forest to capture the complex pattern in RGB value, which we regard as one of the best to distinguish brightness level.
So for the demonstration of image grading classification, firstly we will obtain our dataset from RoboFlow and then pre-processing it using auto orient, resize, and auto adjust contrast using adaptive equalization. And then we will download the dataset into a zip file. After we upload the data set from the zip file to our first Python script, it will extract the RGB color from the dataset using OpenCV function CV to the color PGR to RGB and then store it in a CSV file. In this CSV file, you can see this is the label of each images, and each image also has the RGB value stored in this CSV file. After that, we will run them into a second Python script. This Python script we will extract the dominant color of each images accordingly into the class and also visualize them according to their highest RGB value. So I will show you after the visualization of each RGB stored in the CSV file according to their class, like this for mentah or unripe. So the image that has label mentah will be visualized their dominant color from the CSV value. Like you can see here, there are so many green that's shown here because Highest value of RGB obtained from mentah class is green. So you can see many green and yellow here because of that. After that, we want to like simplify the image classification for each class. So what we do, we will combine or merge the color from the dominant color from each class into a spectrum bar presenting each class. For here, for mentah class, the RGB sorted using red channel is like this. It has a yellowish class, and then this is for the half right class. It has a less yellowish color than the mentah class. And then for matang or right class, fully right, it has a bluish color. Yes. After we apply our spectrum bar, we also have like the code for random forest classifier, which will evaluate our accuracy of images. You can see here that the Matang class or the fully right class has much better score on accuracy scores. It is more distinct than other class because of the bluish color. That's all for demonstration for image learning classification.
Assalamualaikum, my name is Adam Rizman and I will be presenting about the findings for the image grading classification. In this project, we classified watermelon ripeness using a random forest classifier based on color spectrum analysis. Specifically, the red channel from the RGB. Red was chosen because it's closer to the yellowish tones seen in ripening watermelon, while green blends too much with the outer surface. We visualize each red ripeness category as a horizontal spectrum sorted by red intensity. Unripe watermelons show mostly dark green tones across the three-quarters of the spectrum. Half-ripe fruits extend this green region further, indicating minimal ripening. Ripe watermelons, however, show much more variation with lighter yellowish, bluish, and green hues dominating the right two-thirds. Consistent with the findings from EZA, will link lighter ring tones to wax accumulation during ripening. Our random forest model achieved 71% accuracy overall. It performed best on ripe watermelons with a strong F1 score of 0.92. Performance for unripe and half-ripe categories was moderate, showing room for improvement. In summary, combining color analysis and machine learning offers promising results for ripeness detection, especially for identifying fully ripe watermelons.
Hi everyone! In this part of the presentation, I'll walk you through the methodology and the results of our project, which focus on predicting the sweetness level of watermelons using machine learning. We use a linear regression model to estimate watermelon sweetness based on measurable features in the dataset provided in the file. .csv. These features included factors like density, color, and possibly sugar content indicators, which are commonly linked to how sweet a watermelon is. Our process started with data pre-processing, cleaning the dataset, handling any missing values, and analyzing the feature distributions. We visualize correlations between variables to understand which features had the most influence on sweetness. We then split the dataset into training and testing sets and used scikit-learn to train a linear regression model. This algorithm was selected for its simplicity and interpretability, especially useful when we want to understand how each feature contributes to the final prediction. Now for the results. Our model achieved an R-square score, which means it could explain approximately that percentage of the variation in sweetness across the watermelon samples. The mean absolute error, indicating the average difference between predicted and actual sweetness values. From our analysis, we found that density had a strong positive correlation with sweetness, suggesting that denser watermelons tend to be sweeter. This aligns with real-world expectations and validates the model's predictions. In summary, the linear regression model provides a useful and interpretable way to estimate watermelon sweetness, and our results show promising accuracy given the data.
Assalamualaikum warahmatullahi wabarakatuh. Today I will be presenting about the linear regression for in our project for our watermelon sweetness determination. And for this part, we started with a dataset from RoboFlow containing 70 images labeled manis and tidak manis. And using the dataset, we also split it into two with the training set and the test set size. 80% for the training and 20% for the test set. And after that, for modeling, we compared two approaches. The first method is linear regression for comparison between. Then the second method is logistic regression, which is more appropriate for binary classification. Like in this project, we also evaluated some model accuracy and coefficient matrices, classification report, prediction probabilities, class distribution, feature correlation heat map, and yes.
My name is Kashan Kamsin and my metric number is 2316465. So moving next is our disease detection. For this case, we are using convolutional neural network or CNN as our base model, specifically ResNet 18. ResNet 18 is suitable for our dataset because we are only using the small dataset and it comes with ImageNet that can help in detect the general aspect like texture and shapes. Our dataset is watermelon disease recognition that we got from Mendeley data consists of 4 classes. The first one is healthy, and then 3 diseases which are anectros, mosaic virus, and downy mildew. The next one is our technique. For our technique, we are using transfer learning with fine-tuning final layer. This one is to reduce the time consuming for our training. And other than that, we are also using the techniques like random horizontal flip and random rotation to simulate the image, and also the normalization for the better pixels. For training, the data split is 80 for train and 20 for evaluation using Adam Optimizer. And by this model, we obtain accuracy overall 61%, 64% for precision, recall and F1, and 61 for weight.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Akif Azamli Zazmani. The metric number is 2317647. And now I will present to you about the result in discussion and also the code demonstration.
Okay, for the result discussion, firstly, the code will get the qualitative and quantitative metrics so that we can evaluate the model and how good it is. The first one, we have the validation accuracy. This one, the validation accuracy, so that it will tell the model is how much the accuracy for the model. And then we have the plot of confusion matrix, so that it will tell how the model confuse or not between classes of the datasets. And then we have detailed classification report that will tell the recall precision and also the F1 score of the model. And then we also have the plot of training metrics that will tell how the model performance when we train after some epochs. Then we also have the show predictions that will give visual outputs for the model for the prediction on the evaluation dataset. They will tell the disease of the image, whether it's healthy or mosaic virus, anthrax, or downy mildew. And then we also have pre-uploaded image that if we run this, then we can put some random leaf so that the model will predict whether it is one of the disease or healthy. So yeah, then we will run the code. Okay, after we run the code, and as you can see here, after 10 epochs, we get the validation accuracy of 0.6466, that means 64.66%. And then for the coefficient matrix, we get a huge number in the downy mildew. That this is means 89 correct, and only 2 is incorrect for the downy mildew prediction. And then we can see here the healthy one. The model can correctly predict the healthy one, the 35 healthy leaves, and only 3 are incorrect. And we can see there's a confusion between anthracnose and mosaic virus. It is because the model is cannot distinguish between mosaic virus and anthracnose. This might be because of the quality of the picture, or maybe the resolution, or maybe some improvement needs to be done on the model. And for the plot of the training matrix, we can see the training loss decrease over time and the accuracy increase over time. It means that over epochs, more when we get more epochs, the model is improving and becoming more accurate. And for the visual output, we can see, as expected, for the Dundee mildew, the model can correctly predict it, and for the healthy one also we get the same result. But the anthracnose often mispredict as mosaic virus. This might be because of the, as you can see here, the quality of image is a little bit blurry. Okay, and then for the uploaded image, I have run the code, and this is and I upload the anthracnose leaf disease leaf image, and it predicted as downy mildew. So the model is not excellent enough or not good enough to predict an anthracnose disease leaf. And then for the to get the detailed classification report, I have run the code, and then as you can see here, the classification report, the precision, recall, and F1 score reading. The anthracnose one is and the mosaic virus are a little bit low, but the downy mildew is very excellent with um, almost 100%, and the healthy one is 82.22%. And we get the F1 score accuracy of 0.6784. So that means that this model is excellent in certain areas, but it needs an improvement, especially in distinguishing between anthracnose and mosaic virus. Maybe a clear, more um, more clear image and more quality dataset can be used to get the model get better performance. So that's all for the code demonstration. Thank you.
Okay, now we came to the conclusion. So, in the conclusion, this project has shown how useful it is to use AI to assess the quality of watermelon from traditional way like tapping to modern way by using the image detection. With our models, image grading, disease detection, and sweetness detection, the assessment of watermelon quality now only take a short time, even for mass quantity. Not only that, our model also has shown a high accuracy in determining the watermelon quality, indicate that our project is successful. So in the end, we hope that we can use the AI in our daily life as long as it for a good cause. So thank you so much.